Okay, let's give it a start. Once again, hello and welcome. This is our second session of the PML School, where we want to teach you how to build custom PK and PK/PD models in Phoenix non-lin. My name is Bernd Wendt, and together with my colleague Chris Mehl, we will be running this series of webinars and which comprises this school. But before we begin, I would like to point out that if you have any questions during the webcast, you can use the Q&A widget on the right-hand side of the console. We'll try to answer any questions at the end of the seminar, but if we run out of time, we will be answering those questions in a follow-up mail, and you will get those after the meeting. Right now, I want to start with an introduction to the PML School. Although I have presented this in our first session already, I think it's necessary to do this once again since we got a large number of new people registered for this second session. But I want to keep this really short in order to save time for the actual modeling topic of today that Chris will present later on. So the PML School, or better the School for Learning Phoenix Modeling Language, is an idea that is an initiative from the Cetara support team. Together, my colleague Chris Mehl and I, we will be presenting most of the modeling exercises in individual sessions starting from October of this year up to August of 2017. And we are supported by Dan Weiner, who has volunteered to cover specific areas. Just for your information, all the exercises, the materials and models that we present in this series of webinars are taken from the book Pharmacokinetic and Pharmacodynamic Data Analysis from Johan Gabrielsson and Dan Weiner. This is a standard reference for PK/PD modeling. And here we have selected a set of 20 very common scenarios. And these scenarios have been picked on the basis of the frequency that we see those associated support cases. So we have this information that we collected over the last years in support. And we think these models should represent very common scenarios. And since it originates from a support question, we also think these scenarios represent roadblocks for our user base. So we hope that with these sessions that we give here in the PML School, we can steer you around those blocks and provide solutions. Additional information about this book. The book of Johan and Dan has recently been released in its 5th edition. It contains a large theoretical part, but also comes with more than 100 different exercises covering all sorts of modeling scenarios, and they are explained in any detail in this book. The book also comes with a USB stick that contains all the exercises in the form of Phoenix project files. So for our PML School, we have been granted permission by the publisher to present 20 exercises from this book in live seminars, and we can make the models available in text form. So here's the overview of the 20 models that we select. The first one, the 3-compartment model, has already been covered 2 weeks ago by a lecture given by Dan Weiner. Today, Chris Mehl will talk about parent and metabolite kinetics. All the rest of these models will be presented in further sessions as we go along. So this is actually the path that we would like to take when we are presenting our exercises and the models. You might be familiar with this Phoenix model interface. As you see here, the bottom panel representing the built-in options or libraries that you can pick from. We will show you how you can make selections within this built-in options for the different scenarios that we cover. But the built-in options do have a equivalent as a picture, as a graphical model. So it's just a different representation. And we will also show you the options that are available in the graphical model builder, how you can adjust the graphical model to adopt your scenario. And finally, the graphical model as well as the built-in options, selected built-in options, do have another representation that is the textual model, and that is really where we want to go and where we want to end up with. It's also just a different representation of these two other functions, and this is actually what the compute engine in the program, in the software, is actually executing. But by going through these steps, built-in options, graphical model, textual model, you will see how these are related, and you will also see how quickly you can adopt the language that is presented here in the textual model. In most cases of the exercises that we present, the bulk part of the model is selected from the built-in options. In some cases, we use also options provided by the graphical model builder. And finally, when we go to the textual model, there only needs to be some tiny changes, little changes, to really represent the scenarios that we are talking about. So learning of the Phoenix modeling language should become an easy and quick endeavor. That is really our goal here. And at the end, after each event, we will make the materials available in our Certara forum that is shown here, the link. And it will contain the slide deck as well as the model in form of a text file. And these text files can easily be imported into the Phoenix model object, and you can run this on a new data set. We have seen quite some traffic and a few discussions going on the forum after our first event. I would like to further encourage the use of this platform for exchanging questions and comments. And we will provide responses there as soon as we can. But alternatively, you can always, come to us in support and send us an email there or use the web portal. But that's the introduction of the PML school. Now it's time to hand over to Chris Mehl. He will talk about parent and metabolite kinetics. So Chris, please take over from here.
Thanks, Bernd. So today's topic in the PML library school will be PK19. This is from the Gabrielsson and Weiner text reference. And this shows how to do simultaneous modeling of parent and metabolite kinetics, specifically with Michaelis Menten elimination. Okay, so our objective here is to simultaneously fit the parent drug and its metabolite. We have data for both. And we'll model capacity limited elimination, also referred to as Michaelis Menten kinetics. And at the end of the day, the objective is to find estimates for the model parameters, which are VC, which is the volume of the central compartment for the parent compound, VT, volume of the peripheral tissue compartment for the parent, C clearance, this is the intercompartmental distribution rate for the parent compound, the Michaelis Menten constant, Km, and the maximum metabolic rate, Vmax. And then for the metabolite, we will also estimate the first order elimination constant, as well as the volume of distribution for the metabolite. Okay, so the protocol, what was done to collect data. So we only have one subject here, and this subject received IV infusion doses of the drug at different dose levels, 10, 50, and 300 micromoles per kilogram on separate occasions. After each dose administration, concentrations of the drug and metabolite were measured in the blood plasma, and the plasma samples were collected. So we start with an exploratory data analysis. This is a semi-log plot of both the parent and metabolite concentrations by dose group versus time. So we have 10, 50, and 300 micromolar doses administered, and we see the profiles for both parent and metabolite. Notice at the highest dose level, we start to see some separation between parent and metabolite. When we go to actually model the data, the starting point in the Phoenix model object is to use the structure tab to define the model. So the Phoenix model object is a very flexible way of doing modeling. It can accommodate PK, PD, PK-PD, indirect response, and a variety of different types of models that may be of interest. So we start by selecting the model type, which in this case is PK, set the parameterization, what's the input, the dose administration type, number of PK compartments, and so on. And as you make these model selections, the base PML code is written for you and shown in the statements block over here. Whenever you start with a structural model, you will also get a graphical model for the parent compound. The graphical model is a nice flow diagram that shows the different compartments that are involved in the model. And we see in the different compartments, there are labels that show some of the different parameters or inputs to the model. So starting from here, this is the central compartment, blood plasma, and observations are collected from this compartment. This central compartment has a volume of distribution, VC, and this represents the drug input into the system. From the central compartment, the metabolism is modeled using Michaelis Menten elimination with Km and Vm max parameters. And we also have distribution of the parent compound from the central compartment to a peripheral tissue compartment. The concentration in the peripheral compartment is represented by C2, and this compartment has a volume of distribution Vt, and the rate constant for flow between these compartments is this clearance parameter. Okay. So we start by modeling the parent compound alone. This way we can have good confident estimates for the parent compound. And how were these derived? Well, according to the Gabrielsson and Weiner reference, the volume of the central compartment, the initial estimate is 1 liter per kilogram, and that's obtained from the administered dose divided by the concentration at time 0. The tissue compartment volume is also 1 liter per kilogram obtained from previous studies. Clearance rate, 0.1 liters per minute obtained from previous studies. And then to get initial estimates for Km and Vmax, we use non-compartmental analysis where the average clearance across the dose groups using this equation Vmax divided by Km plus Cp average, the average concentration, one can then come up with estimates for both Km and Vmax. And this is discussed in detail in the Gabrielsson and Weiner reference. Okay. There's also an additional very handy tool in the Phoenix model object that allows you to derive initial estimates graphically. So when we're modeling the parent compound, we can take those estimates from Gabrielsson and Weiner, but we can also see an overlay plot. So we start with some observations, and then with these being the model parameters for volume of distribution, Km, Vmax, and clearance, you can modify the values here and see what would be the predicted concentrations given these set values here. And then you can overlay the predicted curves with the observed data to visualize good initial parameter estimates. So this is particularly useful for parameters like Km and Vmax that might not be easy to determine initial estimates. Okay. Once we're satisfied with the model fit for the parent compound only, we'll make a copy of the model and paste it back into the workflow. This retains all of the model settings, structural parameters, and parameter estimates from the previous run. We can then edit the copy of the original model and switch to textual mode and add the metabolite observations and structural parameters to the model. Okay, and here we have the textual model in PK code. This is commented out. So this is broken into segments. The first segment is differential equations for the parent in the central and peripheral compartments. Next, we have the differential equation for the metabolite. So we see that, so for example, for the parent, we have the Michaelis Menten elimination as well as the distribution between the two compartments. And we also have the differential equation for the tissue compartment. And then for the metabolite, we have the rate of formation of the metabolite, which is from the Michaelis Menten part of the equation, minus the elimination of the metabolite, which is the amount of the metabolite times the metabolite elimination rate constant. Okay. Next, we have the central compartment. The concentration in the central compartment at the start of the model is the amount, which is the dose, divided by the volume of distribution for the central compartment. And then we have the administered dose, which is referred to as A1. And since this is an infusion dose, it has a duration of 5 minutes. The peripheral compartment concentration at the start of the model, C2 equals A2 divided by the volume of distribution for the tissue compartment. Next, we have observations. This observed statement talks about the observed concentrations for the parent equals the concentration in the central compartment, and then here we have this error model, where we have a power error model with an epsilon equals 1. For the metabolite concentration, the concentration of metabolite is according to the volume of distribution of the metabolite and the initial amount of the metabolite. And then we have the observed statement here. This is where we're actually comparing the observed to the predicted, and the error term handles differences between predicted and observed. Lastly, we have the PK parameters, which are modeled as fixed effects. And here, so this is the list of all parameters. And the parameter estimates, we basically use a C statement, C, comma, and then this is the lower bound for the parameter set to 0, initial estimate, which is 1, and then the upper bound, the maximum value the parameter can take, which is 5 in this case. Okay. So given that the parent compound has been modeled, what we can do is come up with initial estimates for the parent. I'd like to switch over to the Phoenix application now and model the parent compound, and then we'll come back to modeling the metabolite. Okay, so I'm sharing my screen, and on the screen we should see the Phoenix application, and we have here a overlay plot of the parent by dose and the metabolite by dose versus time. If we look at the input data set, this is the structure of the input data set. Here we have time in minutes, and we have a column that has the concentration of the parent in micromoles per liter, as well as for the metabolite in micromoles per liter. We also have the administered dose. So this is where we record the different dose levels, whether they be 10 micromolar, 50, or 300 micromoles per kilogram. Okay. Okay, so to begin modeling the parent compound, we'll go ahead and send the data set to Phoenix Modeling and then Phoenix Model. I'm going to rename this to parent only. Okay. Okay, and typically it's best to start on the structure tab when defining the model. You may notice that this population checkbox is checked. Typically for individual modeling, we don't need this mode. However, in this case, there's a handy feature of the population mode that allows you to do an overlay plot of all of the different profiles. So we're going to take advantage of that and leave the population mode checked. The type of model will be set to PK, and we will use clearance parameterization. The absorption is intravenous, and we specify that it is an infusion dose by checking this infusions possible checkbox. In the closed form is the integrated solution. I've unchecked that to show the differential equations. Okay. And if you notice over to the right, the statements section will show you the differential equations and the model inputs as well as the observations. So as I'm making these menu selections, the PML is written for me. The number of compartments is 2, and we see that the statements expand to include a second differential equation for the peripheral compartment. And then another specification of this model is that we have Michaelis Menten elimination. And I can simply check this saturating checkbox, and that will add the Michaelis Menten kinetics to the model. Okay, so this is the structural model. And typically what one would do is start with a simple model for the residual error. I'm going to cover what was done in Gabrielsson and Weiner, which was to use the power error model. Which fit best, and here we have power 0.5. So we're simultaneously modeling 2 different sets of data, and this is a useful weighting scheme in this case. Okay. Now that the structural model has been specified, I will go ahead and specify the model variables, which include the time, and that's mapped here to time. And since we're modeling parent only, the CP parent will be the observed concentration for this fit. And then we will use dose for the ID variable. So the dose, this will be used to profile the different curves. Next, I will go ahead and specify the administered dose. Okay, so the administered dose, we will use an internal worksheet. And notice since I use the dose variable to profile the data, I get this nice little reference here that shows me the dose. Now, when we're entering infusion doses, the capital A stands for an amount. So the total amount of the dose is 10. And since this is a 5-minute infusion, since we're getting all of this in 10 minutes, the rate is going to be 2 micromoles per minute. So that will be my rate. And then the time is the actual start of the infusion. So that's going to be 0. For the 50 micromole dose, the amount is 50. The rate will be 10. And the start time is also 0. And then lastly, for the third dose group, 300, and then 60 micromoles per minute, and then start time of 0. Okay. All right, next we'll go ahead and set initial estimates. These are the starting values for the first iteration of the model run. And what I'd like to do here is switch over to the initial estimates tab. This is a very useful feature of Phoenix model objects, where one can see the observed concentrations and then modify parameter estimates to see what the predicted values will look like. So we'll start with the time course of the observations, which is 300 minutes total. I will also do an overlay. The overlay will show all of the data from all of the profiles on a single plot, where the observations are the circles and the predicted is the line. And also we'll show this on a log scale. Might want to toggle back to linear for the time being. Okay. So I will enter initial estimates from Gabrielsson and Weiner, where the volume of distribution was 1.4, and then the Km, we had 15, and then for Vmax, we had 1, and for the clearance, 0.1. Okay. So if I switch to the log scale, I can now see with these initial estimates approximately where the predicted line falls in relation to the observed variables. And if I increase some of the parameters, I can either use the slider bar or I can type over the value. If I increase the Km, I start to get a better fit. And then volume of distribution, for example, I can increase this to, say, 2. Maybe we'll increase the Vmax. And we can see the fit's getting better and better as these values get modified. Now just remember, these are initial estimates. The algorithm will start with these estimates and then go through iterations and fit the final estimates. So I'm going to say that these are good enough for initial estimates and use these little left-facing arrows to copy them to the initial estimates for my model run. Lastly, on the Run Options tab, I happen to have an NLME license. So I go to Help About Phoenix, and I actually have both Win Non-Lin and NLME. I don't want to model in a population mode. I'm only using this because I want to overlay all of my plots. So I'm actually going to switch my method from one of the population models to naive pooled. And in fact, if you have just a Win Non-Lin license, when you go to the population mode, it is set automatically to naive pooled with no other options. So we'll stick with the naive pooled on the run options. And at this point, I'm ready to run the model for the parent. And so here it goes through different iterations. Okay, so the model has completed, and I can look at the final estimates, which are going to appear on the theta worksheet. So again, these were the structural parameters for the parent compound. And now we'll switch over and look at some of the results here. I will want to look at some of these plots. There's a lattice plot. The lattice plot will show the fits for the 10, 50, and 300 micromolar groups. I will switch the y-axis to logarithmic to better visualize the fit. Okay, so that's the lattice plot. And we can also look at the overlay plot, which is the one without the word lattice selected. So I can go ahead and pop this out and, again, view in log scale. Okay. So this is the fit for the parent compound for the different dose levels. At this point, typically one would refine the model and go through different estimates, but for the sake of presentation, we'll say that these are the final estimates and we're happy with these. I'm now going to switch back to the slides and we'll talk about the next steps to simultaneously model parent and metabolite. Okay. Okay, so what we can do is switch over to the text mode. It seems that I've had an issue with the slide here. So I'll go ahead and share back out my Phoenix application. But the point here is that we can use this, this existing model to now model the metabolite as well. So my starting point here is to copy my parent model and paste it back into the workflow. Okay. And so now I have a copy of the model with all of the structural model already set. And we'll go ahead and switch this to parent and metabolite. Okay. Now I should comment that you do have a model representation. This is the graphical user model. And in this context, the graphical user interface is not as useful, but we'll see several examples later on in the PML school series where we actually go into the graphical mode. To model the parent and metabolite simultaneously, I will go ahead and switch the model edit to Textual. And so here we have a text representation of the model. Okay. So I'm going to do a bit of housekeeping here. So one aspect of the Phoenix model object is it expects to do population models. So we have these population EDAs, EDA terms that we don't need, and we also have some random effects, a random effect block that we don't need. So I'm going to start by removing these. Okay. And we also don't need the structural parameters. We're just going to model the parameters as fixed effects. And I'm going to remove the typical value designation for each. Okay. And then rename my parameters as desired, which are volume of the central compartment, volume of the tissue compartment, Km, Vmax, and CLD. Okay. And I'll need to edit my equations accordingly. So wherever I see CL2, I'll replace with CLD. Okay. And also in the concentration portion of the equations. And you might have noticed in the bottom options panel, you might see some text going red. That's a guide to tell you, what the model doesn't understand or something new is needed there. So that's a handy feature. Okay. What I'm going to do next is go ahead. I already have the equation for the central compartment for the parent. What I'm going to do is I'm going to copy this equation and paste it back in, and then this is going to be the equation for the metabolite. Okay. So in this case, for the parent, the Michaelis Menten is the metabolism, the removal of the parent, but for the metabolite, it's the production of the metabolite. So that's why we remove the minus sign here. And I'll use a paren to enclose this statement here. And then instead of minus distributive clearance, since that's not what we're modeling for the metabolite, instead what we're going to model is A0 times the elimination rate of the metabolite, KME. Okay? And then I need a closed paren here. Okay. Now it tells me KME is undefined, so I'll need to go ahead and define KME. I'm actually going to copy the KM and paste it back here, and we'll call it KME. So now that it's defined as a fixed effect, and I've used it, it can be used in this equation here. Okay. For the error model, we have, this is the C-OBS, and to differentiate it from the metabolite, I'm going to call this C-OBS PA for parent. And then we need to be able to accept observations for the metabolite. So I'll just copy that line and then call the second line C-OBS ME for the observed concentrations for metabolite. Okay. Now this C refers to the the concentration of the parent. For the metabolite, this is actually going to be CME, where CME is defined here. CME equals A2 divided by, oh, okay. This is actually, I need to add one more statement to define the concentration of metabolite. So that's CME equals A0 divided by volume of distribution for metabolite. And now that this equation has been entered, I need to define this VME as a fixed effect. So I'm going to copy one of the volume statements from here and add it in the model, and we'll define this as VME. Okay. Okay, so in the context here, we get this warnings tab, and this warnings tab is also very useful. So in this case, it just tells me that the observation columns are not mapped. Okay, so that tells me that I have to go back to the main setup, and I will map the parent concentration to C-OBS parent, and the metabolite concentrations to C-OBS metabolite that were defined in the model. The dosing should still be in place from the previous run. And lastly, I can go to the initial estimates tab. Remember, this is the visualization tool, and we had the initial estimates for the parent compound. And we've actually defined some initial estimates in the textual model. So I can check the overlay here and see if they, whether they make sense. Okay. So under the model parameters, I had actually copied these. So what I want to do is I want to change these 0.15 and 0.3 for fixed effects. Okay. And if I switch back to the initial estimates tab, these actually get pulled in from the model text statements. Okay. So if I'm happy with these, I can now go ahead and run the model. Okay. And we'll start with the plots. So here we have the lattice plot. So this is the lattice plot where we see the different dose levels for parent and metabolite. And then the IVAR plot will show side-by-side plots. So the plot on the left is the metabolite at the 3 different dose levels. And the plot on the right is the parent at 3 different dose levels. And from here, you can also look at the final parameter estimates and see the different final parameters. So, and also look at the CV% to see the confidence in the estimates. Okay. Okay. So this concludes the presentation of the modeling in the Phoenix tool. At this point, I'd like to go ahead and open up for any questions from the Q&A.
Okay, thanks, Chris, for the nice presentation. And we've got an initial question already, but as I mentioned at the beginning, I mean, we are starting now with the Q&A session, and you have the Q&A widget on the right-hand side of the console. Please enter your questions there, and we will try to find an answer or give you an answer right away or as a follow-up after the meeting. So, Chris, there's one question from the audience. For the metabolite A0 is used as a parent drug dose. Should this be adjusted for the molar ratio of the parent and metabolite?
Yeah, so that's a good question. So actually in this case, the A1 is used as the parent drug dose. The A0 is the metabolite starting concentration. So the concentration of the metabolite is A0 divided by the volume of distribution for metabolite. And the A0 is actually derived from this differential equation here. If you look at line number 6 in the slides, and the A0 is derived from the metabolism of the parent compound, and then it's adjusted over time minus the A0 times the elimination rate. So we derive the A0 from the parent using the Michaelis Menten kinetics.
Okay, we've got several questions regarding the error model. I mean, what is the indication here? Is there any indication why you use a power model as a residual error model?
Yeah, so typically what one would do is start with an additive error model and fit the parent, and then see how the fit is, and then if there's a bias, for example, to the tail portion of the curve or the lower concentrations, one can switch to a different model such as a multiplicative. In this case, we are simultaneously modeling 2 different sources of data, and we're only using one error term, 1 C epsilon. But since we're applying that same error term across 2 different sets of observations, the power model can sometimes scale and handle that better. And so in that case, it was chosen. And there's more discussion about this also in the Gabrielsson and Weiner reference.
Another question, can you just point out once again why you use the population mode? because that was just a question here. What would happen if you uncheck that box?
Sure. So if you uncheck the box, and we could do this instead of in population mode, we could do it to individual mode. If I if I share back out my Phoenix application. Okay, so before I make any changes here, I just want to point out that we're getting a nice overlay plot here. We'll switch back to the parent fit. We get this nice comparison with our simultaneous set where we see a nice overlay of all of the different profiles. If I switch back and don't do the population mode, so instead of using dose as an ID variable, I use dose as a sort variable here, okay? And I might need to rebuild my worksheet. Yeah. And so now I will check my initial estimates tab and then just rerun the model in non-population mode. So we just get a slightly different output in this case. In terms of how the graphs will look. Okay, so one difference here is if we do it in non-population mode, we're getting a separate set of parameter estimates for each level of the dose, right? So for dose level 10, we get one set of parameters. For dose level 50, we get a second set of parameters. And for dose level 300, we get a third set of output parameters. And then also in the output plots, we do get the nice overlay, but that's the key difference. Whether we're getting one set of parameters for all of the profiles simultaneously, or whether we're getting one for each sort variable, which in this case is the dose group. Okay. Are there any other questions, Bernd?
Yes, sure. There's one actually interesting question about the PML code. Are the fixed effect statements the only place to enter initial estimates? It would mean having a different model for each individual if one has a study with multiple individuals. So the question is how to model with how to get initial estimates for this kind of scenario. For example, you have 3 different dose groups. Do you get? Will you take into account different initial estimates for each dose group, or is it just one?
Yeah, that's a great question. So actually, we're starting with the same initial estimates for all of the subjects in this case. So even though we got different sets of final estimates for each dose level, we still started with the same initial estimates. And then these are controlled on the parameters tab and then fixed effects subtab. So we see here, and these are all the initial estimates.
Oh, very good. There's one other question about the metabolites. I mean, in this case, you've modeled for just one metabolite, right? From the parent, there's one metabolite. How can you deal with multiple metabolites?
Yeah. So provided there's enough data available, you can certainly deal with multiple metabolites. So you would still start with the base structural model for the parent compound. Apologize for the slow update here. So you would start with the base model for the parent compound as before, And then you would need, in your input data set, you'd need another input column. So you'd need to basically add a column with observations from metabolite 2, if there's a third metabolite for metabolite 3, and then you would have to go back into the model and add the COB statements for each metabolite, as well as at least one differential equation for each metabolite.
Okay, very good. There was several questions asking once again about how you came up with the initial estimates. Maybe you can go back to this slide and explain once again.
Okay. Okay, so the initial estimates were derived from Gabrielsson and Weiner. Bernd, would you be able to maybe go back to the slide? For mine is stuck.
Okay, sure. Oh no, wait a second. Oh, this is stuck for me as well. Let me take over control.
Mm-hmm.
Then I'll go back to the slide. Where are the? Yeah, here. You see that?
Mm-hmm. Yeah. So, so for the parent compound, the initial estimates were the central compartment volume was obtained from dose divided by the concentration at time 0, which is if you use non-compartmental analysis and model the data as a bolus, you can get a C0. And so you can use this as your volume estimate. Or you could also use the volume from the NCA fitting as well, would be another approach. And it says here, obtained from previous studies for the tissue volume and the clearance volume of distribution. This is per the Gabrielsson and Weiner reference. And the KM and Vmax from NCA, basically you took the average clearance across the 3 dose groups, and set them equal to Vmax divided by Km plus the average concentration from each dose group, and then solved back for the Vmax from this equation. It's discussed in Gabrielsson and Weiner, but in the presentation, what I did was I used the initial estimates tab on the Phoenix model object to further adjust these values as initial estimates.
Okay. Yeah, I think that's mainly, we've covered most of the questions right now. We'll get, we'll save all the questions that have come in that we might not have covered right now, and follow up with an email. So thank you for your interactions here. Just as a reminder, I've listed here the next series of events. Our next session will take place on November 3rd, where I will present how to model PK and PD data with repeated oral dosing. This is followed by a seminar held by Chris on November 17th. And on December 1st, it's my turn again by showing you how to simulate oral and IV data separately and simultaneously. The last session of the year is reserved for Dan Weiner, who will present a TMDD model, a model for target-mediated drug disposition. As a reminder, we will post the materials that is a slide deck as well as a textual model on our forum, where we'll also provide a link to the recorded webinar. On behalf of Certara Support, I would like to thank everyone for attending today's presentation. This concludes the webinar. You may all now disconnect. Thank you very much. Thank you, Chris, and bye for now.